while you keep yourself warm with a cocktail, and I hope they make them strong, I will take you to a tour through our immune system, which is a fascinating and very complex organ, with a specific focus tonight on how we can use our immune system to fight cancer. So, see if this works. So our immune system is not just an organ, it's a system, as the words say, because it actually connects multiple different organs that is used to either educate our immune system or to clean it off when it has sort of done the job. It is composed of many different cell types that forms a very complex interaction and signaling together to allow us to keep our bodies safe. So what is done is that this complex system is designed to protect us against either external dangers. They can be bacteria, fungi, or viruses. So, for example, in these days of the coronavirus, we should hopefully have an immune system that are able even to realize this new foreign intruder and fight it as it goes along. But it's also designed to fight internal dangers, and they can be normal cells that change into cancer cells. So actually it's more and more realized that this process probably occurs multiple times through our lives, but it's just not realized. So early stages of a normal cell transferring into a cancer cell will be cleared off by our immune system. But once we reach a state where cancer is clinical evident, obviously the immune system has failed to some extent. So what is the goal of immunotherapy is to sort of regain and reboost our immune system to recognize this danger called cancer. So here we have a cancer lesion, and here there is an organ that is used to develop our immune system. It's a lymph node, and these beautiful flower-like cells are what is called dendritic cells. So they are really key players in activating our immune cells to various foreign intruders, and also cancer cells. So hopefully we can mount an immune response against this cancer, and these immune cells will travel through our body and enter the cancer tissue and clear off the cells. But this sometimes needs help, and that's why we have designed immunotherapy. So the aim of immunotherapy is to boost our immune system, and that's why the two words are connected, immuno and therapy. So the therapy itself is not killing the tumor, but it's activating the immune system, hopefully to do so. So that's kind of fundamentally different from the conventional type of therapies for cancer, such as chemotherapy and radiation, that targets directly the cancer cells. The advantage of this, at least if it works, is that our immune system actually has memory. So it can memorize what it have seen like years ago. That's, for example, why when we once have had an influenza, we are much faster in clearing off a new influenza as it comes along. And the same goes for cancer. So if we have had an immune recognition of our cancer cells and the majority of them are gone, there might be some small lesions still resting in our body. This can be cleared over time by the immune system if it has realized this danger. So there is a certain group of cells in our immune system that is sort of the key players when we talk about cancer and immunotherapy. Those are T-cells. So T-cells, we can also say that they are the elite soldiers of our immune system. And the reason why these are important here is that they have the capacity to kill other cells. And this is what we would like to do with cancer tissue. So, but first, before it kills, of course, it has to recognize that something is wrong and that it's dangerous. And for this, there needs to be some difference compared to the healthy tissue, because we don't want to have our T cells playing around and kill our healthy tissues. So what makes cancer cells different from healthy tissue is that they acquire a lot of DNA damages. That can have various patterns, such as mutations or DNA strand breaks or translocations. But the outcome of any of these changes is that there will be changes in the proteins within the cancer cells. So these proteins, so the red guys up there, once they are degraded, they will be transported to the cell surface and presented here. So this molecular complex here kind of serves as a window where the T cells can look into the cell. And this actually happens in all our cells in the body, not only cancer cells, but also all our healthy cells. So T cells are able to patrol our body to try to snap up if there are anything that looks dangerous. So through this complex that we called MHC class 1, or major histocompatibility antigen 1, we have sort of a way to look into the cancer cell. So we have sort of a lock here. And the T cells carry also a molecular code, namely the T cell receptor, that serves kind of as a key to this lock. So once there is a pair between the T cell and the cancer cell, we have a situation where the T cells can kill the cancer cell. So T cells would release substances that would introduce the killing of this cell. So if this process happens efficiently, then we have a successful therapy, and we have a successful clearing of the problem. So, but this of course needs help. So now I will show you an example of such a therapy where we are boosting T cells in the body. Basically, you can take out tumor lesions. And in those tumor lesions, often there is a number of T cells. It's kind of a numbering game, and they are often not substantially strong, or there are not enough of them to fully clear the cancer. So we can help it along by expanding them in the laboratory and then give these T cells back in large number to the patient, and then clear off the tumor. So this was done some 20 years ago. It was pioneered at the National Institute of Health in the US. And here are two examples of patients from this treatment. So here you can see quite evident a large tumor on the neck of a patient with metastatic melanoma. And this patient received this adoptive cell therapy. So T cells expanded from the tumor, given back to the patient, which completely clears off the lesion. In another example, which is a bit nasty, so that's a warning, there was a very large lesion on this patient neck that was also cleared off by immunotherapy. So these two examples and many others documents that T cells can be really efficient in sort of wiping off the cells that they are targeting. So, so this is what we would like to further achieve. But sometimes when you're developing something, it's also good to have an eye back on what actually happened years ago, because sometimes crazy things was tried in medicine, but sometimes actually we can learn some lessons from that. And I think we can learn a lesson here from this strategy. So use dead bacteria to treat cancer. That seems like a really crude idea. But it was actually an idea that was used. So this gentleman is called William B. Coley, and he lived more than 100 years ago in New York, where he was practicing at a cancer center. So he was actually, probably without knowing, he was the pioneer of immunotherapy. So what he did is that he took a mixture of Streptococcus bacteria and meat extracts, and he injected it directly into tumor lesions. So this mixture is now known as Coleys toxin, and as you can see from New York Times here in 1908, he actually treated over 400 patients, and he probably cured around 150 of them. So this was actually a pretty amazing achievement. although also a number of the patients actually felt very bad after this treatment. So I think that what we can learn here is that we should actually remember some of these success stories and try to find out how can we actually do this today in a more elegant way, not just injecting bacteria into a cancer lesion, but actually allowing our immune system to be alert that there is something dangerous. So I think what we need to think of is that immune therapy today is kind of a reinvention of this Coleys toxin in a more elegant manner. So sort of around 100 years later, that's about 20 years ago, there was a new invention made which really sort of revolutionized the treatment strategies and established cancer therapy as we know it today. And this invention was taken off by these two gentlemen here, and for that they received the Nobel Prize 2 years ago. So what James Allison and Tasuku Honyo did is that they identified certain regulatory mechanisms in our immune system, and by blocking those, they could kind of reintroduce a reaction to cancer cells. So this has been sort of determined as releasing the brakes against cancer. So that can be sort of looked as brakes in a car. So actually you kind of take off either one brake or the other, and thereby allow the immune system itself to reactivate towards cancer. And this actually works very well for many patients, better for some patient categories than other. But it works so well that it has sort of established itself as what is known as the fourth pillar of cancer therapy. So many patients, cancer patients today, will meet the option of receiving immune therapy that was kind of originated by this invention. So now comes a little bit more a molecular picture describing what this brake actually does. So these brakes are also determined checkpoints, so the drugs are called checkpoint inhibitors. So there are kind of two fates that a T cell can have once it is either activated by the dendritic cells or once it meets the cancer cell. Either it can be deactivated and walk away, or it can be activated and kill their target cell. So these checkpoints determines the difference between deactivation or activation. So there are some molecules here that provides a signal to the T cell to be deactivated and walk away. But if you block this signal, you can regain activation of these T cells. So by this strategy, one could allow already established immune responses in a patient to actually make a difference in terms of killing cancer cells. So instead of these cells just sitting in the lymph node doing nothing, they could actually make it to the tumor, infiltrate, and kill cancer. So this is now a strategy that is very often used. It activates the immune system, but it doesn't give any direction. But it works surprisingly well. So this is the data that sort of made this drug to the market back in 2014. This is a plot where each bar here is a patient, and it's the sort of change in the tumor mass from the time point 0 to the end of treatment. So if you have a bar up here, you have an increased tumor volume with therapy, or if your bar is down here, you have a decrease in tumor volume with therapy. So this is known as a waterfall plot. And what you can see here is more than half of the patients do have a decrease in their tumor volume by being treated with this checkpoint blockade strategy. So actually in this patient cohort that was treated here, namely advanced melanoma, this result was so amazing by that time because basically nothing else had worked in the stage of disease where these patients were treated. So it's the first time in history that a drug has been approved by the FDA, which is the medical authorities in US, based on what is known as a phase 2 trial. So of course, with the requirement that larger clinical trials should be conducted, but for ethical reasons, this drug was already given to the market and taken out to patients after this very small testing phase. So it's now used in many different cohorts of cancer patients. But as I said, the problem with this strategy is that it activates our immune system, but it doesn't give any direction towards the cancer cells. So I think what we need to do in the future is to be better at directing our immune system towards cancer, so make what we call precision targeting, and also allow our immune system to better infiltrate tumors. So to go about these strategies, I will tell you a little bit more detail about how T-cells can actually recognize cancer cells and how we have evaluated this in a number of clinical settings. So I mentioned earlier on that changes in cancer cells, being DNA damages, give rise to changes that can also be seen on the cell surface of cancer. It's known from these checkpoint inhibition that actually patients with a high mutational burden does better than patients with a low mutational burden. And this is seen in this case in patients with lung cancer, but also in patients with a number of other cancers. So actually when this data was first realized, there was quite a ethical debate on how people should sort of, how this should be given to the public. Because what it means is that in this case with lung cancer, cigarette smoking induces your mutational burden. So the direct message of this finding were that if you have lung cancer, the first thing you should do is to start smoke, because then you will basically up your mutational burden and you will have a higher chance to respond to immunotherapy. I don't think that this has ever sort of been scientifically tested, so I don't want to encourage you to try that out. But I think that it's just interesting because it's directly different from what we could observe with other types of cancer treatment, where usually many mutations is a bad thing. But with immunotherapy, it's a good thing. And the reason it's a good thing is that it makes the cancer cells look more foreign and more dangerous than those with a lower mutational burden, and therefore they have a higher chance to be realized and seen by our immune system. At least that's kind of the hypothesis, and we want to understand whether we actually have T cells that can recognize these mutations. So to do this, we entered kind of a journey where we needed to develop new technologies to describe this molecular window between the T cell and cancer cells. Because that we didn't really know how these mutational changes would be sort of translated into something that our immune system can actually see. So that has been also the journey that I have taken scientifically. It's been to find new technologies where we could interrogate the interaction between T cells and cancer cells, and how we could identify which targets are recognized by our immune system. So then the reason why this was really a challenge, and is still a challenge, is that cancer is very heterogeneous. So while we are having these DNA damages in cancer cells, they don't appear similar in all cases. So in fact, they appear like completely heterogeneous across different patients. So each cancer have their own mutational signature, and basically no cancers looks the same. So it's only a very, very small fraction, probably less than 1% of the DNA damages that occur, that are actually similar across cancers. All the rest are completely heterogeneous. So we have a situation where every patient that comes in the door and want to have immunotherapy have basically their own type of disease signatures. So that's one level of complexity. The second level of complexity is our immune system. So our immune system is also very different from individual to individual. So our immune system is raised to look into this molecular window that we call MHC class 1. These molecular windows comes in very many different flavors. So we have them inherited either from our father or from our mother, and there are 6 different types of those. So we will end up with 3 from each of our parents, and then have a unique set of those 6. And probably we can't find anyone in this room that have exactly the same 6 molecules. There are, of course, some of them that are more frequent than others, but having a match in all 6 pairs is very unusual. So that gives us basically a combination of 2 very heterogeneous systems, ends up with a massive heterogeneity. So the consequence of this is that every patient have their own immune system and their own type of disease. So that kind of forces us to make what we call personalized therapy. So we have to find basically the rules that determines how our T cells can be tumor reactive, meaning what are the keys that we can identify in a given patient, and what are the locks that allow these immune system T cells to enter into the cancer cells. So to do that, we developed a system where we basically are using nature's own strategy for making a very good coding system. So a very good coding system is our DNA. So DNA can code for a very large variety of things, and it codes for how we all look and feel and appear, which is obviously also different. So DNA only have 4 bases, but the combination of these 4 bases can give a very high number of different barcodes. So this can be determined a DNA barcode, and such a DNA barcode can be used to label also molecules. So what we do is that we build molecules that mimics the cell surface of a cancer cell, and this composed of these molecular windows and the fractions of mutations that are carried along with them. And this complex that mimics the cell surface is then tagged with such a barcode. So barcodes are, as I said, stretches of DNA and can be read like any barcode, as the same as you would see in a supermarket when you go shopping. So basically what we do is we select the cells and then we scan them through a barcode scanner, which in this case is called DNA sequencing. And with this DNA sequencing barcode scanner, we can determine what those T cells are able to recognize. So with this strategy, we can mimic the cell surface of a cancer cell, and we can create thousands of different varieties of this surface to see what targets are our immune system actually able to recognize. But this has to be done for each and every person, and that's of course an experimental, quite heavy task. So what happens is that when we have material from a cancer patient, we can sequence their tumor and identify what mutations are present in that tumor. From this, we can create a map of potential targets that our immune system can see. And from this map of potential target, we can generate the reagents that would mimic the cell surface of a cancer cell. This we will then mix with immune cell from the patient, either immune cells sitting in the tumor or immune cells from peripheral blood. And then we can basically measure what the immune system is capable of seeing. So after a while, we will have an understanding of what are the locks that are present on the cancer cells and what are the keys that we can use to open those. And this is a process that then, once we have enough data, we can use basically new strategies like artificial intelligence or fancy computational models to try to identify similarities between patients. So what is, when we have a certain selection of mutation, what is likely to be the best ones to actually target, for example, for a cancer vaccine or for cell therapy. So this is what we are at the moment is trying to achieve, to have that signature that provide us with the correct targets. So I will show you a few examples where we have done this in patients that have been treated with the immunotherapy. So these slides are a bit more busy, but what we have is, in this case, we have looked into patients with bladder cancer. So these patients have been treated with immune checkpoint inhibition to release their immune system. We've sequenced their tumor, and we have identified what could be the potential sequences that could be present on the cell surface. Then we have predicted through different computational algorithms the likelihood of these sequences to occur on the cell surface. When we have made our selection, we have then built the complexes that should resemble the cell surface, which in this case is then labeled with a DNA barcode. And then we can plate those out and mix them with T-cells. We can then barcode read them through sequencing. So once we are through this process, we may end up with a picture like this. So this is a representative example of a bladder cancer patients that have been treated with the anti-PD-L1s, or a type of checkpoint inhibition. And this particular patient also had a response to therapy. So what we see here is that before treatment is initiated, when we look in the blood of this patient, we find only one T-cell population, being this one, that recognize a mutation that has occurred in the patient cancer. We also find a T-cell response that have recognized a virus that most of us have, which is called Epstein-Barr virus. And then we also screened for a number of other mutational signatures, but these were not recognized by the cancer. But then what happens when we give this checkpoint inhibition to release the immune system is that we start to see T-cell populations occurring that can recognize multiple of these mutational changes in the cancer. So after 3 to 4 weeks, we can see an increased number of T-cell populations, it's all those up here, and we can see that those persist for several months or even years after therapy. So this is kind of a signature of immunological memory. So this immune system here after 3 years, even though the cancer at this point was completely eliminated, still remember the mutational damage that has occurred in this cancer cell. So if one cancer cell would be hidden somewhere, there is at least a hope that these immune cells could clear them off. So if we try to sum up on those patients that we see, there is, as is unfortunately often the case, also a great variation. But we do see a signature that patients that respond to therapy, which is the blue guys up here, after 3 to 4 weeks have an increased number of T-cell population that can recognize their mutational damages, compared to patients that do not respond to therapy. So there is a sign here that we can hopefully, by inducing a larger number of T cells that can recognize mutational changes, actually be able to move the patients from this category over to this category. So another example that I will share with you is a patient cohort with metastatic melanoma, where we basically did the same. So this is the same plot as I described before, with the only difference that this patient group now is patients with metastatic melanoma. So again, we studied for each patient the mutational changes in the cancer and how this can be recognized by the immune system. So this patient cohort comes from the National Center of Cancer Immunotherapy at Herlev Hospital, where patients have been treated not with checkpoint inhibition, but with the therapy I showed earlier on in the presentation. The same therapy that removed those nasty tumors on the neck, was given to this population of patients. So basically a pool of immune cells extracted from patient's tumor and expanded in the laboratory and given back to the patient. So we can see here, again, as an example from one patient that was entering this trial, when they started, they didn't have any recognition of any mutational changes. So the red dots up here represent T-cell population that again recognizes viruses, such as Epstein-Barr or cytomegalovirus, whereas all the blue signatures is recognizing mutational changes. But then patients were given this pool of cell up here. And what is in there? So it's kind of a mysterious product that is just expanded from the tumor, but we don't know what is there. So what we looked at is what is actually in this bag of cells. And here, luckily for this patient, there is a lot of T cells that recognize mutational changes. So all these blue dots here are populations of T cells that recognize DNA damages. And once they are re-injected into circulation, they can be seen and detected for a long time in circulation, and then there is a slow decline, and in the end, after 1-2 years, in this patient, we could no longer detect these T cells. But we can see that when we look across multiple patients for these signatures, we can see that actually this is, again, something that induces the chance of patients to respond to therapy. So we looked at two characteristics. We looked at how many different mutational changes can be seen by our immune system. So can you see one mutational change or can you see 10 mutational changes? That is sort of the breadth of this immune response. And here we can see that patients that have a good response to therapy, either what is known as a complete or partial response, the yellow or the pink guy up here also have a long time stable disease, the green ones. They tend to have many different T-cell population recognizing mutational changes. At the same time, it's also important what is the size of these populations. So how many T cells do we have that circulates that can recognize cancer cells? So numbers also play a role here. So this is the number of T cells that are present, and that also seems to correlate with the outcome of therapy for these patients. So I think what we can learn from these studies is that what we actually knew, hypothesized, knowing that a large tumor mutational burden was actually predicting a good response to therapy, we can now actually document that T cells can also recognize these changes, and also the presence of such T cells are important for patients' outcome. So this is another investigation from the same set of patients, and again looking more into the magnitude of the T-cell response, so how many T-cells are actually there. We can still see that patients that do good have more than those that don't, and they also tend to have a better overall survival if they have a high number of T-cells. So what we can learn from these strategies is that we can basically try to design two things. I think we can try to design strategies where we can make, let's say, early diagnostic tools, or not necessarily diagnostic tools, but at least early detection tools, whether patients actually respond the way we would like them to respond to therapy, and therefore hopefully are ending up in the category that does good, something like this. Or if they don't, if they're not able to mount a immune response early on, they would likely end up here, and we should immediately do something different, not waiting to the time of death, but actually try to offer them another treatment strategy already very early on. So I think those are the tools that are now trying to be developed in the field, so that we can basically tailor the therapy and direct it very early on to the patient's need. Another lesson that we can learn is that you really need a high number of T cells. So there are, of course, multiple strategies where you can achieve such T cells, and that's, I think, is something that we may need to add, at least to patients that cannot mount this response themselves. So one strategy that we are currently working on is this, where we are taking peripheral blood, and then we can activate and expand tumor-specific T cells while culturing them in the lab and then give them back to the patients. So it's kind of similar to what I showed before with adoptive cell therapy, but in this case, it differs in the term that we can take the T cells from peripheral blood and we can activate them directly towards the target that we know is present on the surface of cancer. So what we make use of is to mimic how T cells are expanded and activated in our immune system in a natural situation when we, for example, have a viral infection. So here, the T cell would need to have 3 different signals from this dendritic cell that sits in a lymph node. So it will present the molecules of danger to the T cell that would then be fully activated, and you can see it will retrieve what we call a license to kill signal and capacity, and can go out throughout the body and find any foreign and dangerous cell that looks like having this same window that the T cell was educated to see. So again, we can build a structure that would mimic this surface, and we can then provide it with all the same signal as the dendritic cell would do. So with these correct signals for activation, we can mount a larger army of T cells that are ready to fight cancer and have the functional capacity to kill the cells once it meets this dangerous cell in any other site of their body. So this is just one example of many different strategies that are developed to boost our immune system to recognize cancer. So I would say that overall there is a horizon where there is new strategies emerging for cancer immune therapy. And in this horizon, you see now the sun has turned into an antigen-specific motif, either with a certain T cell receptor or a certain antibody on the surface that are ready to specifically interact with cancer cells and kill only those. So let's hope that we can have many more of these suns circulating, and that will be of benefit for future patients. So last but certainly not least, it's just really been a pleasure to be here today and share with you the insight to our immune system and how it can fight cancer. It's also really a pleasure for me to go to work every day and work with a fantastic research group where the majority of them are up here. Because this is, when you do science today, it's really a team effort. It's not only sort of a gray-haired old man looking through a lot of large books, but there's a lot of things that we need to do in larger groups with different expertises, and it's a pleasure, certainly, to work in this team. We can also not do any science without funding, so a great thanks to all the foundations that supports our research so that we can move forward and hopefully solve the puzzle of immune recognition of cancer. Thank you for listening tonight, and enjoy the cocktails later on.
Thank you so much.
Thank you. Bye.